This is Selma Schmoll at the Multidisciplinary Cancer Congress 2011 in Stockholm. Professor Dr. Hans-Joachim Schmoll, thank you for taking time to join us during this multidisciplinary meeting here in Stockholm. And you are certainly one of Europe's leading physicians in the area of GI oncology. And I'm hoping you can talk to us a little bit about what we're learning at this meeting in regards to the GI cancers, and in particular, colorectal cancers.
Thank you. It's a great pleasure to being here. This is a meeting which is not showing in GI cancer brand new results of very big trials, but a lot of consolidation of in-between results and some new results from trials which have been recently finished, as well as a lot of preclinical data, genomics and biology. The most interesting probably are new data from the colon cancer area, colorectal cancer and advanced stages. Today was a nice report from the Spanish trial on advanced colorectal cancer, where maintenance treatment after induction chemotherapy was investigated whether the anti-angiogenic agent bevacizumab would be sufficient instead of continuing chemotherapy. And they looked on the KRAS expression and potential differences, and they found out that indeed KRAS mutated tumors, so patients with KRAS mutations in the tumor, had a worse outcome under this chemotherapy. It was Celox plus bevacizumab than those with KRAS wild type. There have been several reports before, contradictory results, and this was now a very nice study with many patients favoring the prognostic value of KRAS wild type versus mutant. So this is a piece of information which is more relevant for the future. In particular, they investigated also circulating tumor cells, which is another biologic parameter for prognosis under treatment of advanced colorectal cancer.
Let me ask you a question, because there has definitely been a shift in the way we're treating this cancer from the days where we were very limited to purely chemotherapy. Could you maybe take us through a little bit of the evolution from where we were to where we are, and what is the state of genomics and the biology and the molecular characteristics that we understand now about colon cancer?
Years ago, many years ago, there was only one drug available, which was given all patients independent of biological or molecular subtype that was not known. And even with the addition of different chemotherapeutic agents, all patients have got the same. So now we know that at least one parameter, which is the KRAS expression for wild type or mutation in the tumor, makes a difference for those patients who are going to get the EGF receptor antibodies, cetuximab or panitumumab. In addition to this molecular marker, there are many other molecular markers which can be identified, which are separating different biology of the tumors. But still we do not really know how to use them. Some of them are with contradictory results, but in the next 2, 3, 4, 5 years, we will have many more subgroups in colorectal cancer, which is a BRAF mutation subgroup, and so on and so on. Currently, and shown today again, it was clearly shown from the Spanish group that circulating tumor cells, together with the mutation of this gene RAS, is an excellent indicator to put patients into different prognostic categories. That means based on the clinic, but based on the RAS mutation and the circulating tumor cells in the blood before beginning the treatment, can identify the patient very, very precisely in a group with a much better outcome or less outcome or intermediate outcome. So this is the first time that we have a biological score to guide treatment decision. It is still too early to make this as routine, but it's now showing that we are on the track using these biological and molecular options to make patient definition for better, more precisely and adequate treatment.
Personalized medicine.
Yes.
Does that mean you are now combining the classic chemotherapy with these new biologic agents?
That we have done before as well, but we had to treat most patients without selecting them. Now we could probably better define which patient has more and who has less benefit and just only toxicity of this combination.
How do you test, let's say, for one of these mutations like the KRAS?
This is done by the pathology in the tumor tissue which has been taken out on the biopsy. It's a standardized method which most experienced pathologists in the world now can do reliably. Circulating tumor cells, for example, can be done also by a commercial available test which is doing well and reliable. So more and more these genetic analyses and biological additional tools can be done in the clinical routine. However, they're more expensive, they are not prospectively validated, so it takes time to put that in a clinical reality.
How many mutations have been identified to date for colorectal cancers?
Those of clinical relevance with respect to prognosis and potentially to select treatment, these are about 5 today, but we know there are many, many more, and probably many more highly relevant. And in addition, we means all the clinical researchers and preclinical laboratories and companies look on gene signatures, which probably more precisely and easily can combine all these factors in a single gene array and saying you are in a very high risk, you should deserve that treatment. So colon cancer is on that way, but it takes probably 5-8 years to have arrived, which is on the stage which is for the patient beneficial.
So what is the gold standard today? If the patient, is every patient in Europe now being tested for some kind of mutation before treatment is initiated?
Oh yes, because the registration label for the EGFR inhibitors like cetuximab or panitumumab requires this testing before, but there's only for KRAS. For the other mutations, it's still experimental or done in a clinical research setting, not in a routine.
And if I recall, last time we talked, it was like FOLFOX, or was that not the standard of care?
No, it's much more differentiated what is the best first-line treatment for the patient, and there was a European consensus which we have just finalized, the first consensus describing what is the best treatment for a given patient in this given situation. And there are all the options listed and also recommendations are given what is better for that situation and worse. So the first time we have a uniform definition what should be or could be the best for a patient in this personal clinical situation.
May I ask you what you're doing in your lab? What are you doing research-wise?
We look on the combination of new drugs and look what is the best working in the different tumor types, in particular in different biology of colorectal cancer cell lines, to go then for the clinic with these combinations in the moment, but also we look on other biological regulators of the disease.
And I also imagine that the clinical trials arena is very global in this area, the United States and Europe, that these, and Asia, these are global clinical trials.
Most of these trials are very global. For example, I just finished as chairman of the trial the international colorectal cancer trial looking for the better treatment and more cure. This is 1,100 patients in several parts of the world, from Australia to many parts of Europe, and all of these trials are now very international. Another trial which I finished together with a company was going from Vietnam to Canada. So all of these trials need the international cooperation to collect the patients in a short time, because so many drugs, so many new options are there and they need to be evaluated, and we need all patients to do this, and the patients mostly have benefit because they're part of a new treatment option which could help them.
Thank you, Professor Hans-Joachim Schmoll, who comes to us from Germany.
Thank you.